Hi, so my name is Alex Clare, and as was said, I work at Hitachi Chemical Advanced Therapeutic Solutions, and this is a partnership between ourselves and Distech to bridge some gaps in the T-cell manufacturing process, where we see there's some issues with current bioreactor culture platforms, and I'll get into some of the work we did there. I'll give an introduction to HCATS and Distech, and I'll take a look at the current state of manufacturing and why we see this gap and why we're trying to address it. And then investigating new culture methods. I put that in quotes basically because cell therapy has a fairly negative view of stirred-tank bioreactors and really look at only either static flasks or rocking bioreactors as a viable solution. But these are really established culture methods from biologics. And then I'll wrap up. So Hitachi Chemical Advanced Therapeutic Solutions is a subsidiary of Hitachi Chemical. I'm based out of the Allendale site, and we have another one on the West Coast in Mountain View, as well as a partner sites in Munich, Germany, and Yokohama. So we see ourselves as helping clients deliver products and create a world in which transformative therapies are accessible to all. And what that means is we have to figure out solutions where the incentives might not align for our clients to invest their time and money in that. So we engaged with Distech because they have a really strong business case as a single-use and functionally closed bioreactor, and it also gives us the flexibility with some of the features to both customize the control regime and mimic the internal human environment a little bit better with some of the gas mixing. So the current state of CAR-T and TCR manufacturing, if you guys are familiar with this image produced by Wells Fargo, it's looking at all of the T-cell based immunotherapies that are currently in clinical trials. And what we're seeing is a lot of therapies that are at the preclinical or phase 1 stage and are progressing through pivotal trials into BLA very quickly. And they're encountering issues with the manufacturing process available to them, where they're not able to consistently produce in specification product. And this is getting into kind of an engineering problem for these companies, where the method of action for T-cell therapies is better understood, and we know that these are effective in treating at least leukemia-based cancers. And we're evolving into what's an engineering challenge, which is adding control and capability into manufacturing processes in order to give patients access to these therapies. And capability in this sense is actually a term for the ability to produce in specification product. And here's a quote from the CEO of Novartis about how they're really not able to achieve that with the manufacturing technologies available to them today. So they're looking at solutions as they expand indications and as the biological imperative becomes more clear. So when we were looking at which challenges to tackle first, we had a few considerations, and I highlighted the COGS analysis because it makes a nice picture and you can visualize it very easily, but our optimization efforts needed to affect both manufacturing reliability while also making those therapies more accessible to patients by reducing cost and reducing cost of goods. So we wanted to target a unit operation that is more ubiquitous to T-cell therapies, which means we could have looked at cell engineering or the way the CAR or the TCR is injected to cells, but that is pretty variable across the industry. And if we were to try and optimize viral vector transfection, for instance, we wouldn't be able to affect change in electroporated T-cell therapies. And so we were looking at expansion because it both represents a high cost aspect of the manufacturing, and the vast majority of processes that we look at do require some extent of cell expansion to meet dose criteria. So now we have to ask the question, why are we using the culture platforms we currently use? And if you look around the industry, you see the G-Rex as a viable static flask culture technique. You see a lot of companies engaging with GE on the Zuri, and now you see some application of the Clinimax Prodigy as a fully closed and automated system. But we had to ask why stirred tank bioreactors are so adopted by other industries for suspension cell culture, but not in cell therapy. And the big answer we got was sheer stress. So we engaged in some small-scale studies that we felt went well enough to scale that up to full autologous scale, and this is what we ended up with. We used the Distech as our culture platform, and we integrated it with the Lovo spinning membrane filtration system to allow us to have continuous perfusion in the later stages of culture. So we actually just sterilely welded that onto the device and used the Distech controller to operate both devices. And looking at kind of the technical specs of our culture, we control dissolved oxygen to 50%, pH to 7.15 by sparged air and CO2, as well as we had a baseline. And then we had a media formulation of Tex-Mex media supplemented with human AB serum and Pluronic and Antifoam to allow us to sparge. We actually tried this with overlay, but the oxygen demand was too high. So you can see on the left some of our small-scale experiments, and it answered the question, can you culture T cells in stirred tanks? And it was a resounding yes. We saw really rapid cell expansion and consistent at both low and high media exchange rates. So we took those parameters and translated that onto the 2-liter Distech system. And you can see here, runs 1 and 2 are performed with a donor, and then 3 and 4 are performed with a second donor. And the growth curves within each donor collection are actually very similar, which is encouraging because typically the donor collection, even within a donor collection, you're getting variable culture expansion rates. So especially between through day 6 or 7, we have very high correlation within a donor set. And we're also getting high expansion rates, so population doublings exceeding four population doublings within a week of culture. And so we were happy with that, but again, people are going to ask the question, how does this compare to conventional methods? So we took a look at the Distech bioreactor system to the Zuri rocking bioreactor, and we got log increases in cell expansion, and we got more control over the expansion rate and the culture parameters within that culture period. So the big thing for us here is being able to allow therapies to meet dose earlier in the culture period, so you're actually reducing the entire manufacturing process, as well as achieving higher expansion rates with lower media utilization. So this data isn't shown here, but what the graph on the left actually indicates is that we're getting twice as much cell expansion per milliliter of culture media in the Distech system than we were in the rocking bioreactor, which kind of logically makes sense if you think about it from a fluid dynamics point of view. Your flux is going to be much higher in a stirred tank bioreactor than it is in a rocking bioreactor. The same reason people are afraid to go into a perceived high shear environment. And What it works out to be actually is the power input, which is a surrogate measurement for shear stress, is comparable between the rocking bioreactor and the stir tank system. So you're getting a higher mass transfer rate at a similar power input, and without an increase in shear stress to the cells. And so on the right, we're looking at lactate concentration and LDH concentration. So LDH is a qualitative measure of cell lysis or cell death. And early in the culture, we see very comparable LDH values between the rocking bioreactor and the stir tank. So that indicates to us that we're not causing any undue stress on the cells with impelled media mixing. However, as the lactate rates grew, we had increases in our LDH concentration. So we could actually increase our population doublings by having a more aggressive perfusion scheme. So we could actually reduce the manufacturing timeline even more from what now would be a reduction of about 24 to 48 hours to potentially 72 or 96. And that's just by increasing a pump speed. And you'll probably end up using about the same media over the shorter period of time. And so if you take these advantages and looking at how it both affects the reliability of your process and again the cost, which both of those play into patients being able to access your therapy, you see 8% savings on the total cost of goods just from switching from a rocking bioreactor to a stirred tank, with less optimization than you're going to do for your specific therapy. And so the increasing efficacy of the culture reduces the requisite culture period, which is then going to reduce your labor costs and reduce the amount of media you consume. And process improvements are going to be agnostic to what your therapy is targeting. What sort of genetic engineering technique you use, whether that's lentivirus, adenovirus, some non-viral permanent transduction method, and how your cells are selected. Kind of putting that all together, in order to increase patient access to what are proven to be revolutionary T-cell immunotherapies, the industry needs advanced manufacturing methods that increase process capability. And stir tank bioreactors, as an established culture method in biologics and other industries, provide a distinct advantage over rocking bioreactors in both total yield and efficiency. So the BioOne sub integrated with some sort of media exchange device provides a viable culture system for T cells. And in this case, we use the LOVO, but you could also use just a simple hollow fiber filtration system that would require no other equipment that you could just run with the peristaltic pumps on the device. If you want more information on the BioOne system, you can go visit Distech at booth 3904. And thank you.
Alex, thank you very much. Quick question from the audience. We have time for one if anybody's. Okay, great. Oh, hold on one second.
I think you might have answered that right at the end. I was trying to figure out, you mentioned perfusion many times and then CSTR as being the bioreactor. So I was trying to figure out why you called it perfusion.
So the LOVO spinning membrane system, we ran continuous perfusion through the peristaltic pumps. So
I think you, so the question is, and I think you might have answered that, what is actually the perfusion device? You mentioned...
Oh, yeah. So if you see on the right, the LOVO, so that is a spinning membrane filtration device that was designed for platelet washing or downstream formulation. So we actually had them build a program that deactivated their peristaltic pumps and their waste scales and just left the spinning membrane active. So that spins at like a 1200 RPM. And then using the Distech peristaltic pumps, we can run a positive pressure through that filter. So I guess getting a little more technical, but the way the spinning membrane works, it actually creates vortices that prevent the cells from contacting the membrane. So the cells are unaffected by the filtration, but you force spent media through the filter.
Yeah. Thank you.
Yeah.
Great, thank you very much. Yep.
Hi there. Yeah, I'm from GE Healthcare, and I was just wondering how did you compare your agitation and rates between an SDR and a rocking bioreactor? Because to me that seems like there might be a disconnect there. And also your perfusion rates, were they comparable between the rocking bioreactor as well as the SDR, or were they not?
Yeah, so the perfusion conditions were kept the same. So at certain cell concentrations, we increased the perfusion rate from 0 up to 100%. And both on the Zuri and the stir tank. And the rocking rate was used from optimized parameters that we have from other manufacturing processes. So again, it's hard to arithmetically determine the power input of a rocking bioreactor without, a CFD algorithm. So we had to use what have been optimized previously and were shown to work for T-cell cultures. But if we have if there is some equation out there that I can calculate power input, then I could match it up, or I could show that there's mixing and at a lower power input.